Hey everyone, welcome to Reading Recap 6. Is that 6? Yeah, that's 6 fingers. Just double checking. I could have got that wrong. Okay, so for today, we're starting off at page 413. What's really cool about the story on 413 is basically the experience of a graduate student when they made a discovery and got to share their work with some senior people in the field, like their scientific idols. There's nothing you need to know for the exam, but it's a nice story, so I would suggest that you read it. Okay, turning the page. We are now to page 414, basically get an introduction to mutations. So for the most part, everything before 12.1 is really an introductory kind of like context overview, saying that making sure we repair mutations is going to be very important. And it doesn't matter how much energy it costs, the cell needs to do it because it's like life or death. Okay. Now, 12.1, types of mutations. As you go through section 12.1, it is very important that you know every single type of mutation. So transition versus transversion, silent, missense, nonsense. Know what they mean. This is going to be rote memorization. Like, get it down, stick it into your head. What is a point mutation? Turning the page, you go to 416. You see small insertions and deletion mutations. Together, these are called indels. Know what insertion is, know what deletion is. Understand if you remove one or two base pairs, you do a frame shift mutation. Like, what is a frame shift mutation? Let's see, what else is important here? Oh, and then also like page 417, there's an example of a mutation that actually causes expansions of triplets. So repeating codon. So I do want you to focus on page 415 all the way to 417. So we turn to page 418. There's not really anything on this page really to know. Just finish off the reading for the paragraph. Okay, so now we're going to go ahead and skip to page 420. This is another pretty chunky section. And so we talk about the different ways that DNA can get mutated. And I want you to know these different ways. We're going to touch on them in class. The first one that we're going to talk about is deamination, which is caused by hydrolysis. This figure, where's the video? 12.8. Oh, I'm failing. There we go. This video 12.8. You can see all my little notes, like underlining and putting in like details. Know what deamination is. Have in your notes really easily accessible what base something would turn to if it was deaminated. For existence, like know that cytosine gets deaminated to uracil, adenine to hypoxanthine. And then after they get deaminated, know what these products would actually pair with in DNA. So for example, hypoxanthine pairs with cytosine, xanthine pairs with cytosine. So you don't have to memorize these, but I would highly suggest making them easily accessible in your notes. Think we can hit. Okay. So in this section on page 421, it has an explanation in the reading about how you would actually get a mutation that ends up becoming heritable. And then we're going to go over the idea. We will have a question in the worksheet for discussion section that wants you to think through how do we actually get a mutation that becomes heritable if it doesn't get fixed. And really you can only get a mutation if for some reason replication makes a mistake, and after the mistake, it doesn't fix it, and then it gets replicated until we can inherit it with daughter cells and daughter cells and daughter cells. Okay. So next important things, on page 422, they talk about some other different ways that DNA can get damaged, talk about reactive oxygen species. So this is oxidative damage, and there's a couple of examples of those. You don't necessarily need to focus on these, but know that these are ways or things that can cause damage to DNA. There's another thing called alkylation. So that's page 423. Another way that DNA can get damaged. So understanding what alkylation does to a nucleotide and then how it looks after. Once again, just make sure like these types of things, on page 423, they are accessible in your notes. Okay. Now, the bulk of what I want you to focus on is going to be starting from page 423, going all the way to 425, start ending with DNA damaging agents using cancer chemotherapy. This section in the reading goes over the Ames test. We are going to go over the Ames test in class. how to think about it, understand how the Ames test works. The same thing I said, like Hershey and Chase, and for every other experiment that we've gone through, know this experiment like the back of your hand. Figure 1213 is going to be your best friend. You'll actually have figure 1213 in your slides in class. Hopefully you can annotate if you have questions. Oh, I will see you at office hours. You can also ask the LAs during discussion section. There's a whole section about DNA damaging agents in chemotherapy and cancer. The concept that I would want you to know from this reading is essentially that in order to cause death to a cell through I guess a carcinogen or something that will cause DNA damage, the cell has to be replicating. And if the cells are replicating, then you technically shouldn't impact it. Bottom of 425, we have some information about UV radiation and then how UV actually damages DNA. This is by cross-linking. So what I would like you to focus on is going to be figure 12.5, and it talks about making pyrimidine dimers. And essentially these pyrimidine dimers stall DNA replication machinery, which means that you have to fix this mistake. Don't fix the mistake, then the cell can't replicate its DNA. Of course, you can't do that. You're going to end up causing cell death over time. Other figure I want you to pay attention to is figure 1216. This is talking about DNA damage where you can either have a nick in one strain or breaks on both sides or both strands. A one-sided nick is easy to fix. The replication fork is okay. You have a template, it's not a problem, just use ligase. But then if you have an issue where you have breaks on both sides of the strand, you have to use either homologous or non-homologous recombination. We will go over that on Thursday in lecture. And then lastly, we finally start getting into some mechanisms of DNA repair. Today, we're only going to cover, mismatch repair. This is on page 429. And so it's important for you to understand the different types of repair pathways, how they work, and what types of DNA damage they actually fix. Mismatch repair is usually pretty good for those, I guess, mismatches, like those one base pairs that are inserted incorrectly. And then, so what we have for mismatch repair is essentially how bacteria or prokaryotes do this whole entire mechanism. They use a combination of like mut S and mut L and different mut proteins in order to recognize the parent strand from the daughter strand, or recognize which one is being newly synthesized versus which one should be the actual template that we're trying to copy and get correct. And so what I suggest is that you don't need to know all the different like mut proteins and the details of the mechanism. And what I do want you to know is how overall as a concept this repair pathway is working. So for example, methylase, you methylate your DNA, but then a newly synthesized strand doesn't necessarily have the methylations because it takes a little bit longer. There's a delay before it gets methylated. So then you have proteins, the mut family of proteins that are able to recognize whether or not this one is methylated or not methylated. And that's how to determine which one should be the template, which is not the template. And then figure 1217 has little steps by which this mut system can actually recognize the mismatch, which is because there's a deformation of the DNA because there's a mismatch. And then once it recognizes the mismatch, figures out which one's template, which one's daughter. And then once it knows, it basically uses an exonuclease, cuts out a whole bunch of base pairs until it finds the one that was wrong and then refills it back in, usually polymerase 3. So hopefully that will help guide you in your reading and understanding for today's lecture. And then on Thursday, we're going to focus a lot on mechanisms of DNA repair. And so today, focusing on mutations, how do we get the mutations, what is the outcomes of different types of mutations in order for that to end up as a heritable mistake in our DNA. And then lastly, today we're focusing on mismatch repair, and we'll focus on other types of repair on Thursday. Hopefully this helps, and I'll see you later in class. Bye.